Hi, so welcome everyone to the March session of MycoTalks, organised by the MRC Centre for Medical Mycology at the University of Exeter. And you can see the organising committee on the screen there. So today we're delighted to have two speakers join us, Rebecca Shapiro from the University of Guelph and Guilhem Janbon from Institut Pasteur. So the MycoTalks has been running for 5 years now. We try to represent a diversity of individuals, a career stage, gender, geography, and also a diversity of topics. And what we're going to have, as usual, is two 30-minute talks, followed by 30 minutes of in-depth discussion. So we'd ask you to listen to both of the speakers and to submit your questions through the Q&A. And please start your question with an indication as to which speaker the question is addressed to. And then we will ask those questions following the second speaker. And please remember that all the recordings will be available on the MRC YouTube channel, and all the previous recordings are there if you're interested in looking at them. So the MycoTalks runs approximately every month. So in April, we'll be hearing from Runhui Liu from East China University. And in May from Linqi Wang, Runhui Liu in April and Anna Alastrui. And then in May from Linqi Wang and Heather Conti. And as well as MycoTalks, we also have MycoClinics. So these run quarterly, and these are particularly for people interested in the clinical applications. And they present cases highlighting challenges in diagnosis and management of fungal disease. So the next one of these is due on the 5th of June, will be held on 5th of June. And if you're interested in attending, you can follow the link that will be available in the chat. Okay, so today I would like to welcome our first speaker, Rebecca Shapiro. So Rebecca is currently an associate professor in molecular and cellular biology at the University of Guelph. She's an expert in fungal pathogenesis and fungal genomics. She got her BSc from McGill University and then her PhD from the University of Toronto. And following a stay in the US, she has returned to Canada, where she started her own group. So we're very much looking forward to hearing from Rebecca, who will be telling us about her recent work and CRISPR methods. So Rebecca, the floor is yours.
Great. Thank you so much, Geraldine. I'm assuming everyone is seeing my slides, though I don't see the little green box
Yes,
around
that
it.
looks fine.
Okay, great, excellent. Okay, let me just get a laser. Nope. Pointer, if I can quickly. Okay, wonderful. Nope. All right, that's fine. Well, thank you so much for the introduction, and I'm delighted to have the opportunity to be here today. I've really enjoyed tuning into the MycoTalks over the last couple of years, so I'm grateful to be able to have this platform as well. And so today I'm going to talk about sort of a new project, or something I haven't talked about previously from my lab, where we're developing and applying a new CRISPR activation system that we're using to identify genes involved in trisomy-associated drug resistance in the fungal pathogen Candida albicans. And so, like many others, I think in this audience, my lab is really interested in understanding the mechanisms of drug resistance, and we're particularly interested in studying Candida species and primarily Candida albicans. And since a major focus of the work in my group to date has been on the development sort of new genetic and functional genomic approaches, CRISPR-based methods, we've been really interested in trying to apply some of the new techniques that we're developing as sort of new ways to think about studying drug resistance in Candida species. And the sort of facet of drug resistance that we were really interested in tackling in this project was the role of gene copy number variation that plays in drug resistance. And I'm taking this figure from a review that came out just very recently, so very timely for this, that sort of highlights some of the different ways in which increased gene copy number or overexpression of different components of the fungal genome can lead to increased resistance to antifungals. And so I think we can imagine a lot of these scenarios here. Things like overexpression of the target of the antifungal, of course, will lead to increased resistance to that drug. Overexpression of efflux pump systems will, of course, allow the cell to sort of better tolerate treatment with the drug in question. And we can think of other things like increasing certain metabolic pathways or certain stress responses can also help the cell sort of better tolerate treatment with an antifungal drug. And so this is a fairly well described phenomenon. And this review actually went sort of further to dig into the literature and the sort of reports of this kind of gene copy number variation and implication in drug susceptibility. And we see that in particular with Candida albicans, this is a really well documented phenomenon. And we know that there's well kind of established cases of overexpression of drug target or efflux or other mechanisms of kind of increased gene dosage implicated in resistance, as well as many other sort of unknown mechanisms. So this is sort of an important phenomenon associated with drug resistance. And we also know from sort of many years and many beautiful studies from many different groups that there's many different ways in which the fungal genome, again, here with a focus on Candida albicans, sort of amplifies different parts of its genome in order to cause this sort of overexpression phenomenon that can ultimately be tied with drug resistance. And so we know that both either sort of smaller regional amplifications all the way up to sort of entire chromosome amplifications, so trisomies in the case of Candida albicans, are highly associated with drug resistance, and in particular, resistance to azole antifungals, though not exclusively. And so I'm showing sort of two examples of this from different papers, though again, the literature sort of is filled with lots of examples of this. In both cases, we're looking at chromosome plots, so looking sort of across each of the chromosomes in the Candida albicans genome and looking at sort of relative gene copy number. And the top is work from Anna Selmecki's lab from a couple of years ago, looking at these sort of smaller regional amplifications that we see, upwards of very high gene copy number in some cases, which itself is associated with increased MIC, so increased resistance to antifungals. And at the bottom here, the green are highlighting chromosome trisomies, again, so in kind of entire chromosome amplifications also associated with increased resistance to antifungals. And again, there's many examples of this, and we've kind of seen this throughout the literature. And one of probably the best characterized examples of this phenomenon in Candida, of this idea of sort of increased gene copy number kind of associated with drug resistance, is that of an amplification of chromosome 5 in Candida. And in particular, amplification of the L arm of chromosome 5. And so we know from really beautiful work from Judy Berman's lab from many years ago that amplification of the L arm of chromosome 5 through the formation of these different types of isochromosomes leads to increased resistance to azole antifungals. And subsequent work from their lab further showed, when we sort of zoom in on what genes are present on the L arm of chromosome 5, we find that two really important regulators of drug resistance, ERG11, which encodes for the target of the azole antifungals, and TAC1, which encodes for a transcriptional regulator that regulates efflux of antifungals, are both present on this, this arm of this chromosome. So we sort of can infer and understand that increasing the copy number of both the target and this transcriptional regulator can increase resistance to these drugs. And sure enough, they find that as we increase the total number of copies of both ERG11 and TAC1, we see sort of proportional increase in MIC to fluconazole, so more resistance to fluconazole as we increase these genes. And this is sort of a really nice kind of example of these phenomenon that I was describing earlier. And in fact, sort of is a beautiful example of two of these kind of mechanisms by which increased dosage can increase resistance, where we're looking at both overexpression of the drug target, ERG11 in this case, and overexpression of efflux via this transcriptional regulator TAC1. So this is a really well-characterized example where we can sort of understand how this extra copy of part of this chromosome confers resistance. But I want to highlight that there's a lot of other examples of these sort of genomic amplifications, either trisomies or other copy number variants, where the mechanisms that underscore their association with resistance are really less well understood, where we don't really know what genes are sort of implicated in resistance associated with overexpression. And so that's where this project sort of sets out. And really, all of this was initiated a couple of years ago when I was at a conference with two colleagues, Jules and Lisa Gerstein. who were looking at the phenomenon of antifungal heteroresistance and antifungal tolerance, respectively. And then both were discussing that they have identified, in many cases, trisomy of chromosome R as being sort of frequently identified in their heteroresistant or tolerant populations and sort of associated with these drug resistance phenotypes. And I'll just show some of the examples that have been published in the literature since then, kind of indicating this. And so this on the left is work from Elisa Gerstein's lab, where she had evolved posaconazole resistance or tolerance in Candida strains and found many of her tolerant isolates that have trisomies of chromosome R. And work on the right from Judy Berman's group, where also among, in this case, fluconazole tolerant strains, I found a really high frequency of chromosome R trisomies. So we know that it can be an extra copy of chromosome R, this amplification of chromosome R is highly associated with drug tolerance and resistance, but we don't actually know what gene or genes present on this chromosome are really kind of conferring these resistance or tolerance phenotypes. And so this was the kind of the catalyst for this project. And what we were hoping to do is develop functional genomic strategies that will help us to decipher the mechanisms associated with trisomy-mediated antifungal drug resistance, and in this case, in particular, as it pertains to chromosome R. And so, like I said, we know that retaining an extra copy of chromosome R is highly associated with tolerance or resistance to antifungals. And so we infer that increased copy number of one or probably multiple genes on this chromosome is helping to confer resistance, but we don't know which. And our goal is to develop a scalable genetic overexpression system to try to mimic trisomy-associated gene dosage increase. So a system that will allow us to individually overexpress each of the different genes on this chromosome in order to identify genes that, when overexpressed, sort of confer a resistance or tolerance phenotype, and to do this on a chromosome-wide scale. So this was ultimately the goal of this work. And so the first step in this then was to develop a system that we could use to overexpress genes in the Candida genome, and again, do this in a really efficient and scalable way so that ultimately we could move this up to an entire chromosome-wide level. And so this is work that's been spearheaded by a PhD student in the lab, Nick Gervais. I'll also highlight work from Leticia Maroc, a postdoc, who was a previous postdoc in the lab, working on developing similar systems in Candida glabrata. And so the system that both Nick and Leticia developed in Candida albicans and glabrata respectively is a CRISPR activation or CRISPR-A system that allows for this sort of efficient, scalable genetic overexpression in these species. And so the way that the CRISPR-A system works, it's based on sort of a canonical CRISPR-Cas9 system, but what we do is we generate a nuclease dead or D-Cas9 version of the Cas9 endonuclease, which has lost the ability to, it's lost its nuclease function, and so does not cause double-strand breaks in the genome as kind of classic Cas9 does. And so what we do is we inactivate nuclease of Cas9, we then fuse it to a transcriptional activator complex. In this case, we're using this tripartite activator complex called the Viper complex, which are just 3 strong transcriptional activators. And so now we have this dCas9 Viper CRISPR-A sort of activation system. And what we can do is use a single guide RNA to target the entire complex to the promoter region of a gene of interest. And by doing so, we can essentially sort of force driving the level of high level of expression of transcriptional activation or overexpression from any target gene of interest. And so we developed these systems in Candida albicans and glabrata respectively. And really the key to the system is that anytime we want to target any new gene in the genome for overexpression, all we have to do is swap out the targeting guide RNA, which is a short oligonucleotide, and this can allow us to target any gene for overexpression. And so I'll run through how we've sort of validated this system and then how we're moving towards using this to study the chromosome R trisomy. So the first step that Nick did when he was trying to validate this overexpression system was really just confirm that we could, in fact, kind of drive high levels of overexpression and recapitulate known phenotypes of genes of interest in the Candida albicans genome. So Nick designed a couple of different guide RNAs to target the promoter region of the kind of well-characterized adhesin factor ALS1. He uses QRT-PCR so that we can monitor changes in transcript abundance, and finds that depending on sort of where we target the system in the promoter region, we can drive quite high levels of overexpression of this transcript, anywhere from about 5 to 25-fold overexpression. We also want to make sure we can sort of recapitulate phenotypes known to be associated with overexpression. And so since ALS1 is an adhesin known to be involved in biofilm formation, we further looked at biofilm formation in vitro. There was kind of further follow-up work that was done with this in vivo as well, and find that driving overexpression of ALS1 also leads to increased biofilm formation. So we feel sort of confident that the CRISPR system can work to overexpress genes and sort of recapitulate some of these known phenotypes. The other important thing that we learned from the CRISPR overexpression system was that in many cases, not all cases, we find that CRISPR overexpression can correlate with phenotypic outcomes. And this is work that Leticia did in Candida glabrata, again, with a sort of very similar CRISPR system. And what she did is design a number of different guide RNAs targeting the CRISPR system to the promoter region of the sort of well-characterized transcription factor, EFG1. And so this is just showing the relative position of 6 different guide RNAs relative to the transcription start site. And as we sort of expect, we see very variable levels of transcriptional overexpression or not overexpression in some cases, depending on where we target the CRISPR system. So we get different levels of EFG1 expression. We then went on to perform biofilm assays with these mutants, since in glabrata, we know that EFG1 is an important regulator of biofilm formation. And when she correlated these phenotypes, found that the level of EFG1 overexpression correlates really closely with the level of biofilm formation that we see. And so I like this as sort of reminiscent actually of the correlation I showed earlier between ERG11 and TAC1 overexpression and azole resistance. And right, it just sort of demonstrates that we can use this system sort of titrating, looking at different levels of overexpression and correlating this with important biological phenotypes. Also sort of shows us that, as we know, depending on where we target the guide RNAs, we'll achieve different levels of overexpression of our genes of interest. And then the sort of final proof of concept that I want to highlight is that, ultimately the goal here was to look at drug resistance phenotypes associated with trisomies. And so the last sort of proof of concept that Nick did here was to overexpress a well-known regulator of antifungal drug resistance, CDR1, which is an efflux pump known to be involved in azole resistance. Again, targets at the different regions of the promoter, drive variable levels of overexpression of this gene. And then when he monitors susceptibility to the azole antifungal in a minimum inhibitory concentration assay here, where in this case, we're looking at green as sort of growth of the strain and black representing no growth. We see that as we increase concentrations of azole, we get increasing resistance based, pretty closely proportionally on the level of overexpression of CDR1. So we've shown that we can use this CRISPR system to really efficiently overexpress genes and study phenotypes, including drug resistance, which is ultimately what we're sort of gearing up to here. And so the next step was to take this system where to date we had only kind of individually overexpressed a handful of genes and scale this up to a chromosome-wide level. And so again, the goal here was to work with this trisomic chromosome, chromosome R, design guide RNAs targeting every single one of the genes on this chromosome. There's about 1,000 open reading frames. Take these guide RNAs, clone them into our CRISPR plasmid, and ultimately generate a large pool of mutants where we have overexpression of each of the different genes on chromosome, so that we can ultimately study their sort of relative role in drug resistance. And just to highlight that all of this work that I'm going to show is currently unpublished and is work that's been ongoing by Nick Gervais in the lab. Two things that I'll just sort of mention here. One is that all these sort of libraries, as we're building them up, are in a pooled format. So all of our strains are sort of in a single culture, and we're looking at their relative growth. And then secondly is that we use the single guide RNA that's present on our plasmid, which is in our mutant strains, as sort of a unique molecular barcode that allows us to identify what each of the different individual mutants is. So that's what allows us to sort of work in this pooled format. Okay, so this is ultimately the goal, and now I'm going to run through sort of how we actually built up this library and are screening it to look at azole susceptibility. So while this project is being led by Nick, I wanted to give sort of an important shout out here to another PhD student in the lab, Lauren Wensing, who really tirelessly worked to optimize a protocol here by which we can generate very large libraries of CRISPR mutants. And she's been doing this with a CRISPR interference system, just sort of an inactivation version of CRISPR-A, but we're using the same protocols to develop our chromosome R libraries. Okay, and so what we did here to generate the chromosome R overexpression library, like I said, we designed guide RNAs targeting each of the approximately 1,000 genes on the chromosome R. We like to design multiple guides per gene wherever possible. Again, sort of from the previous work I showed, since we get variable levels of overexpression depending on where we target them. And so we design these large guide libraries. So this can be thousands or tens of thousands or more guides. And then we have them synthesized in a pool. We then sort of en masse clone these guide RNA libraries into our CRISPR-A plasmid backbone. We maxi prep up these plasmids, so we have kind of a large pool of CRISPR-A plasmids targeting all the different genes on chromosome R. We then transform this again en masse into Candida albicans. We select at every sort of stage of selection, we select for very large number of mutants to ensure that we get adequate representation of all of our mutants in our pool. And ultimately, what we're left with is this large pool where we have, again, kind of depending, thousands of different overexpression mutants, each targeting a different gene on the chromosome for overexpression. And so Nick has sort of gone through these steps and has ultimately been able to generate this chromosome R overexpression library, where we have representation upwards of about 98% of the approximately 1,000 genes on the genome. With the CRISPR system, there'll always be some genes that can't be targeted, but we're pretty happy with being able to target about 98% of the genes on this chromosome with at least one guide RNA. Like I said, we try to use multiple guide RNAs where possible. So we also have a very large percentage, about 3/4 of the chromosome is being targeted by 6 or more guide RNAs. And this ultimately leads us to a fairly large kind of mutant overexpression pool of about 5,000 mutants, each targeting a different gene for overexpression, and about 6 non-targeting control strains. So in this case, we basically create like a random scrambled guide RNA that shouldn't target anywhere in the genome. We have a bunch of these, and these act as our sort of controls internally in our pool. Okay, so once Nick was able to generate this pooled library, the goal was now to screen it in the presence of antifungal drugs and azole drugs to see which genes, when overexpressed, would increase in abundance in the presence of these drugs, since this is what we would expect of a gene kind of implicated in trisomy-mediated resistance. So we grow these pools up in replicates. We leave them either untreated or treated with fluconazole. Nick has done this a couple of different times at a couple of different concentrations. I'll show sort of one example of this. And then we extract total genomic DNA from these different pools, and we do barcode sequencing over the guide RNA using next-gen sequencing to look at the relative abundance of the different guide RNAs in our different populations, which tells us which mutants are present at lower or higher abundance in our different conditions. And so again, ultimately, we're trying to find these overexpression mutants that have increased relative abundance when treated with the azole fluconazole. So which ones have sort of better fitness, better ability to grow in the presence of these drugs. Okay, and so like I said, we've done this sort of a couple of different times in a couple of different ways. So I'm showing kind of one example of what some of this data looks like in this volcano plot here, where we're looking at the log 2-fold change and p-value of our strains when grown in the presence of fluconazole, of our mutant pool, I should say. And so each dot is showing a different mutant from this pooled library. And what we're showing in the right top quadrant here in red are mutant strains that, when overexpressed, are increasing significantly in abundance in the presence of drug. So these are the hits that we're interested in following through on, that are showing sort of increased ability to grow in the presence of the antifungal. In this particular screen here, we identified about 15 mutant strains. In some cases, we identify more than one guide for the same gene, which is always great. It shows that the system is sort of working well. And from this, Nick has followed up on 9 individual mutants. Again, we've sort of repeated this under different conditions. And so through kind of looking at different screens, we've kind of narrowed in on a subset of these, on 9 individual genes that we're now interested in following up on to look at their role in tolerance and resistance. I'll highlight that one of the limitations of this sort of pooled screening approach is that we can't individually extract any mutant that we're interested in. So when we have these hits that we want to follow through on, we actually have to remake them as individual strains so that we can look at the effect of overexpressing sort of each gene uniquely. And so that's what Nick has done. He created 9 individual overexpression mutants from these screens that we've now been following up on, and I'll show you some of that work. And so one of the things that we were kind of most interested in following through on with these mutants was looking at the tolerance of these strains. As you'll remember from those plots that I showed, the 2 that showed trisomies of chromosome R, these trisomies were really found abundantly in strains that were found to be have increased tolerance to either posaconazole or fluconazole. So we wanted to measure tolerance of these individual overexpression strains. And this is work from Judy Berman's lab, again, highlighting how we can look at the supra-MIC growth as a measure of tolerance. So we're looking essentially at sort of a lower level of kind of trailing growth, in some cases upwards of very high concentrations of antifungals that grow above the MIC level. And so this is what ultimately we're sort of using to look at tolerance of these individual overexpression mutants that we've created. And just as a sort of preview of what some of that looks like, I'm showing here our control strain, as well as 5 of the 9 hits from the pool that we followed up on. These are 5 hits that did in fact show this sort of increased trailing growth, as you can see, sort of similar to the cartoon, where we're looking at sort of increased tolerance and growth above the MIC to growth in fluconazole compared to a control strain. And we then use this sort of supra-MIC growth, this SMG calculation, in order to actually sort of quantify the tolerance levels of these and other hits from our pool. And so when we put everything together, it looks something like this. So what I'm showing here is this sort of measure of tolerance, the supra-MIC growth of these sort of a range of different strains. The green here are our control strains. So these are sort of scrambled guide RNAs that aren't targeting anything. We can think of these as wild type strains. We're comparing them in red to chromosome R trisomic strains. And then in blue are the 9 different hits that we followed up on of individual overexpression mutants from our screen. I'm showing what the genes are down here. So 4 of these sort of did not show significantly increased tolerance compared to control strains. But 5 of them, those 5 that I sort of previewed on the slide previous to this, show significantly increased tolerance to growth in azole antifungals compared to the control. So we're really excited that we've sort of been able to use the screening platform to successfully identify these genes that, when overexpressed, lead to an increase in tolerance. So there's 2 things that I kind of want to highlight now here, and things that we're continuing to follow up on. So one is, what are these genes, right? Of course, we want to know what these factors are in chromosome R that, when increase in gene copy number, lead to this increased tolerance phenotype. And we're actively following up on this. 2 of these are largely uncharacterized. We don't know a lot about them. We don't have sort of obvious orthologs and Saccharomyces to tell us more about them. So I won't say much more about these for now. 2 of them actually have a really similar function. 2 of them are predicted Rab family GTPases, which we are very well characterized in Saccharomyces, and we know to be involved in vesicle-mediated exocytosis. Not a lot is known about these in Candida specifically, but we think this is pretty cool that we've identified two factors working sort of towards the same process very closely, that both seem to show this increased tolerance phenotype when overexpressed. And so we're interested in following up on this. And then finally is HMO1, which is a predicted transcription factor, which has been shown in Candida to bind upstream of genes involved in ergosterol metabolism. So obviously this gives us sort of a clear hypothesis as to how we think overexpression of this factor could well be involved in tolerance or resistance to azoles. And again, we're sort of actively following up on these factors to try to better characterize how changes in abundance of these different genes can be implicated in tolerance and resistance. And then the other thing that we see from this, and sort of maybe unsurprisingly, is that while we see increased tolerance of these individual overexpression mutants, it doesn't reach the level of tolerance that we see of the chromosome R trisomic strains. And this suggests to us, again, probably unsurprisingly, that probably there are multiple factors that may be required in combination for this sort of trisomy-associated tolerance phenotype that we see. And so just very quickly, with the kind of couple minutes I have left, I want to highlight how we're trying to tackle this question and see how we can start to understand how combinations of factors and combinations, for instance, of these 5 hits from our screen may be involved in tolerance. And so for this, we're turning back to sort of our CRISPR playbook and trying to develop kind of yet new systems that we can use to address this question. And so what Nick has been developing is a multi-gene overexpression system for Candida albicans based on the use of another Cas protein, Cas12. And so Cas12 is sort of really well exploited and characterized in the literature, has not previously been used in Candida albicans. And so we've been adapting a Cas12 system for use in Candida. There's a couple of advantages to Cas12 that we want to use for multi-gene overexpression. One is that it targets a different CAM sequence compared to Cas9. This allows us to target certain regions in the genome that may be inaccessible by Cas9. So this is sort of one advantage. But the other major advantage, kind of relevant to this project in particular, is that Cas12 uses much smaller guide RNAs than Cas9. And it has an intrinsic ability to process its own guide RNA arrays, which means that we can provide the cell with multiple kind of sequential guides separated just by a very short repeat region. And the cell can process each of these guides, or the Cas12 can process each of these guides and allows us to simultaneously target different genes in the genome for, in this case, overexpression when we fuse our Viper system. So what Nick has done is develop this Cas12, Cas12 CRISPR-A overexpression system for use in Candida albicans, and is validating its capacity for multi-gene overexpression. And I'll just show how we're validating this. So again, the first step was really to make sure that we could use the system to drive overexpression of genes of interest involved in resistance. So we went back to CDR1 as this well-characterized resistance regulator in Candida. We show that we can use the Cas12 CRISPR-A to overexpress this gene and then see corresponding increases in resistance to fluconazole. And then the next important step was to show that we can use this to efficiently multiplex genes and look at overexpression of multiple genes simultaneously. And so here what Nick did was develop a plasmid that had guide RNAs targeting both CDR1 as well as ERG11 together. And then looked at MIC values of using Cas12 to overexpress ERG11 or CDR1 individually as well as together in this sort of multiplexed plasmid system. And what he finds is that compared to a control strain, we can see increased resistance to fluconazole when we overexpress either CDR1 or ERG11 individually, and then even further increase in resistance when we overexpress them together using this system. So we're really excited and sort of confident in our ability to use the system to combine multiple genes simultaneously for overexpression, and more than 2 as well. So we're just showing kind of this example, but potentially being able to combine multiple of our different hits to look at their combined role in resistance. So what we're currently working on, or Nick's currently working on, is making multiplexed overexpression of these various CRISPR A chromosome R hits to try to look at the combined function of these different genes in mediating antifungal drug tolerance. And we think this is going to really help us understand the relative kind of role of single and multiple genes when amplified on this chromosome is being implicated in drug tolerance and resistance. And so with that, to just conclude some of the kind of main points from this work, we've been developing and applying a novel dCas9-based CRISPR-A system as an important tool for genetic overexpression in Candida albicans. So we've validated this with many different phenotypes. And we've now kind of developed strategies to scale this CRISPR overexpression system and use pool screening techniques to uncover totally novel or previously uncharacterized regulators of drug tolerance that we think are involved in chromosome R trisomy-mediated drug resistance. And finally, are continually developing sort of new CRISPR-based tools, in this case, a Cas12-based tool that's going to, I think, really allow us to easily create multi-gene overexpression strains, and again, try to validate the sort of combined role of multiple factors in mediating these important phenotypes. And so with that, I want to thank everyone in the lab who contributed to this work. I highlighted in particular mostly work done by Nick Gervais, as well as a lot of the protocols developed by Lauren Wensing in the lab, also work by Leticia as well, who's not present in this photo. We've had sort of various sources of funding for this over the years. It's been sort of a really exciting project to be engaged in, and thank you all for listening.
Thank you very much, Rebecca, for a really brilliant, scintillating talk. It's my pleasure to introduce our next MycoTalks speaker, my long-term friend, colleague, and collaborator, Guilhem Janbon from the Institut Pasteur. Guilhem is a vibrant member of the medical mycology community, and his research program on fungal RNA biology and microvesicles is both illuminating and paradigmatic. Guilhem is also serving as the chair of the Department of Mycology at the Pasteur. Welcome to MycoTalks. Take it away, Guilhem.
Thank you, Joe. Let me share my slide. Is it okay like that?
Yes, it's good.
Okay. So thank you very much for this introduction, Joe. So today I'm going to speak about the Cryptococcus transcriptome and what we are doing with it in my lab. Oops, that is stuck. Okay, Cryptococcus. Okay, first few words about Cryptococcus. That is, as you know, a very different yeast as compared to Saccharomyces cerevisiae, and we study Cryptococcus mostly because it's a major fungal pathogen. It has been recognized within the critical group of important human fungal pathogen by the WHO in 2022. And the cryptococcosis, that is the disease caused by Cryptococcus, is causing 200,000 deaths per year each in the world. So Cryptococcus is a yeast. If you look at it in the microscope, you won't see much of difference between this yeast and Saccharomyces cerevisiae. But if you mix this yeast with Indian ink, then because the Indian ink particle cannot penetrate the capsule, it will really reveal this white halo around the cells, which is the polysaccharide capsule. They have another representation here by electron microscopy, and this capsule is a major fungal, this major valence factor from Cryptococcus together with the melanin production, its ability to grow within the macrophage and the production of some enzyme. Cryptococcus is living in the environment mostly, and you can find it in the soil, in the decaying woods, in bird excreta. Actually, the birds are dispersing this yeast around the earth, and we are supposed to acquire it very early in life. It might cause a very minor fever when we are young, and then the yeast stay as dormant, controlled by the immunity, until the immunity doesn't work because of some disease or other problem like AIDS, and then the spore, the yeast are able to multiply, to disseminate to different organs, including the central nervous system, in which it causes meningoencephalitis, which are deadly, in 100% of the time in absence of treatment, and still kill 30% of the patient when the best treatment are put in place. In 2015, different species of Cryptococcus have been recognized. The main one is Cryptococcus neoformans, and this has composed a large majority of clinical isolates. There's a sibling species named Cryptococcus neoformans, and a complex of species of named the Gattii complex, in which you can find the Tau Gattii, Decay Gattii, Bacillus boris Gattii, and Tetra Gattii. In my lab, we are working mostly with Cryptococcus neoformans, but we are also validating some of our results with Cryptococcus neoformans and sometimes with the Tau Gattii. My lab is mostly interested in the transcriptome structure and its variability according to the different environment. And you understand that Cryptococcus is exposed to very different environment outside and within the human body. And we try to understand how this disease is able to answer. We look at the quantity of RNA per cell, for example. We need the quality and the diversity of molecules of RNA that are produced in different conditions. We looked at within the cells, but more recently we have been interested also on the quality of RNA outside of the cells, and mostly the one associated with extracellular vesicles. I'm not going to speak about this part today. I'm going to concentrate on the cellular transcriptome. Over the year, we have produced large data set of RNA sequence, and with those RNA-seq, mostly we have been able to annotate the genome of Cryptococcus neoformans, Cryptococcus neoformans, and Cryptococcus deuterogatii, identifying roughly 7,000 genes in those species. And the main information we obtained from this annotation, that's the richness of those genes in entrons. Nearly all the genes have entrons in Cryptococcus, and the number of entrons is huge. There is nearly 41,000 entrons in Cryptococcus neoformans. These entrons are necessary for gene expression in most of the time, and then participate in the control of the genome integrity in Cryptococcus. Of course, we have studied alternative splicing and show that it was very common, but it doesn't seem to impact to have a great influence on proteome diversity. It's more a means to regulate gene expression in Cryptococcus. More recently, we have produced TSS-seq and 3' TR-seq, as you see in this slide. So TSS-seq is a specific sequencing that identifies the beginning of the transcription, and 3' TR-seq is a sequencing that identifies the end of transcription. And you see that those in both cases, the results are blurry. There is multiple position, and we wanted to know how to use this position, how to position exactly the beginning of the gene. And it was very difficult to do because sometimes, if you look at an IGV, for example, you have two groups of peaks here, and if you look on linear scale, you would have these things suggesting that the beginning of the transcript of the gene is here. But you look in log phase, you will have multiple position, and you don't know whether the position is here, or here. So to solve this problem, we adopt a clustering strategy in which we cluster each of those beginning of transcription when they are closer than 50 base pair one from each other. When there's a gap of more than 50 base pair, then we make another cluster. So in this example, we start with this and transform this in that. So we have here different clusters. So from there, we wanted to annotate the beginning of the gene. So first we set up, we produce a long TSS. Basically, we took the most distal position of the TSS or the poly A in each condition and generate the longest gene as possible. And then because some of these positions look a bit doubtful, we only conserve the one that represent at least 10% of the identified position for a given gene in at least one growth condition. We did that in many conditions. I forget to say that we produce this data in many different conditions. Overall, we obtain 87% of the gene. We obtain a position of the both UTR those were validated by one TSS and one PA cluster, respectively. So we went up with this typical structure of the coding of a coding gene in Cryptococcus. We have a transcript leader sequence of 5' ETR sequence. It is about 177 nucleotide, and a 3' ETR sequence that is about 186 nucleotide. And today I'm going to focus a bit more about this leader sequence. And this leader sequence, sorry to forget to say that this leader sequence contains introns that we knew about it, and the 3' ETR also contain introns. But the most spectacular observation we made was that in this transcript leader sequence, we observe a large number of additional potential translation start codon. Actually, we obtained 10,286 upstream ATG in 242 genes, which is highly different from what observed in Saccharomyces cerevisiae, for example, where only roughly 100 upstream ATG were observed in only 500 genes. So the question was how Cryptococcus neoformans is able to make the difference between the annotated one, the one that is coding for this the protein that is known, from the one that is upstream in the transcript leader sequence. For that, we have to come back to the model of what is a translation, which is a very simplified model, but basically there is a pre-initiation complex that scans the 5' ETR or TL sequence until you find an ATG, and then start to translate the protein. But if you have an upstream ATG in the sequence, then there is different situation. It can only transcribe this UOF, okay, upstream open reading frame, and in this case, there is no translation of the main OF, and probably a degradation of the main transcript that by the nonsense mediated mRNA decay, because there is a premature stop codon here. There is an intermediary solution where there is this leaky scanning model where both the UOF and the main OF are transcribed. So basically, part of the ribosomes are translating the UOF, and part of the ribosome are translating the main OF. So there is a kind of attenuation of the translation of the main OF, a kind of regulation of it. And another model is the translation of both OF, or reinitiation of translation, but I'm not going to speak about it today. So in the case of Cryptococcus, we are able to show that these things exist. Actually, with the help of Ethan Madani that produces ribosec data, we have been able to show that in some cases, for example, in this one, only the UOF is translated. So here you have RNA-seq, you have a pretty even coverage on this gene, but you see that only the part that is corresponding to the UOF is translated. The part that is corresponding to the main OF is poorly or not translated, suggesting that indeed the translation of the UOF is preventing the translation of the main one. And actually, if you look on the genome and look at the gene that contain UOF, do not contain UOF, and look at the one that contain UOF or more or several UOF, then you will see that the ribosome occupancy or the translation efficiency is reduced as much as you get UOF in the transcript leader sequence. So now we know that this UOF is important. We don't know what is the mechanism by which Cryptococcus neoformans is able to make the difference between this ATG to this ATG. So there is two major mechanisms. It's not the only one, but those are two major. The one is the size of the transcript leader sequence. So basically, if the ATG, upstream ATG, is very close to the annotated ATG, then this upstream ATG is supposed to be transparent. And that has been shown in Saccharomyces cerevisiae and in mammals. And we exactly observe the same thing in Cryptococcus. Basically, the ATG that are less than 20 nucleotide upstream of the annotated ATG have no influence on translation efficiency, and we prove that I have no time to show this data. But there is another parameter is the motif associated with the translation codon, and that is named by according to the name of Marilyn Kozak, that demonstrate that using monkey cells in the 80s. And we observe the same thing. If we look at the motif associated with annotated ATG in the 5 most expressed genes, you see this motif that look very, very much as a Kozak motif. It's not exactly the one we found in mammals, not exactly the one we find in cerevisiae, but it's very similar. And actually, if you can weight the quality of this motif and compare the weight of the strengths of this motif between the annotated HG, the one that are just downstream of the annotated HG that they are I named DUG, or the one that are in associated with UOF, you see that the one that are associated with the most expressed gene average strength weight. The one that are associated with the annotated HG as a good weight as well, but the one that are associated with the UOF or the downstream HG are less confirmed, are less good motifs, and so the quality of the motif impact the usage of these ATGs. So basically, in my example here, the ATG has a very good motif and prevent the usage of this one. But if this upstream ATG was not a good one, that it will not prevent the usage of the main ORF. We have been interested also to compare the quality of the downstream ATG and the annotated ATG. Actually, you see that some of them from the downstream ATG have very good score. And actually, if you compare one gene by gene, the quality of the downstream ATG and the quality of the annotated ATG, you have a group of genes for which a downstream ATG has a better score than the one that the annotated ATG. And actually, what was interesting is that in this case, 70% of the gene have the DATG in frame with ATG without any stop codon in between. And for 38% of them, they have a mitochondrial motif separating those two ATGs. That's an example of it. Here you have the RNA-seq and the ribose-seq. You have an annotated ATG that have a score that is 0.7 roughly, and the downstream ATG has a score of 0.9. And you see that the RNA-seq has a pretty even coverage, but for the ribose-seq, you see that coverage is much better at the DATG than the ATG. And actually, in this case, there is a mitochondrial targeting motif. So we can figure out that when the downstream ATG is used, then the protein is going to the cytoplasm. But when the upstream ATG is used, then the annotated ATG is used, and the protein is going to the mitochondria. So the quality of the motif is impacting the proteome diversity as well. So the conclusion from this part is that the TL sequence of 5' are very rich in UOF, and the structure of this TL sequence regulate both gene expression and protein diversity. This is not unique to Cryptococcus. Actually, Saccharomyces cerevisiae is here the exception. Okay, you see that Saccharomyces cerevisiae has a very short 5' or TL sequence, and actually has a very low number in HGG per nucleotide within those TL, and this number is higher in Pombe and in Rospora than in cerevisiae. The second part of my talk would be of alternative TSS usage, because when we did this TSS-seq analysis, what we figure out immediately that the number of clusters we observe or we count in the genome was much higher than the number of genes. We have 23,000 of cluster to 38,000 a cluster of TSS cluster in Cryptococcus, when we have only 7,000 genes. Most of the clusters are in 5' or TL sequence, but we found some that are also in the CDS. And actually, if you look carefully, and some example, you will see that, for example, at this locus, you will have two TSSs, depending on the condition. One is used in stationary phase, and one is used in exponential phase. And you see that the coverage of RNA is pretty even in stationary phase, when you have the upstream exon are roughly expressed at the same level as the downstream one, but in exponential phase in red, you see that the upstream or the exon are less expressed, suggesting that actually there's two transcripts that are made, one in stationary phase, which is long, this is a blue one in exponential phase that is short, what is a red one. And we validate that by qRT-PCR, but also by Northern, and you see that here that you have a long transcript in stationary phase and a short transcript in exponential phase. So we wanted to study that more carefully and more genome-wide, and we have a very naive idea at the beginning to use the same strategies that people use to compare gene expression. So basically, to construct a GFF, to have the position of each the TSS, and then to compare the expression of this TSS according to the condition, and to identify TSS that were more expressed in one condition than the others. And so the idea was to merge those things. So we had the 4 conditions: exponential phase 30 degrees, exponential phase 37 degrees, stationary phase, and stationary phase 37 degrees. And we wanted to merge it. Unfortunately, when we did that, we obtained first a large number of clusters, and because most of them were very small, they were only 1 base pair long, and also we obtained some that were very large, that were more than 100 base long, which was not, to our mind, biologically relevant. So we decided to change the way we cluster this data, and for that she first tried to identify what was a genuine TSS in Cryptococcus, and she looked only at the 50% most expressed gene in exponential phase 30 degrees, and the one that contained only 1 single TSS cluster in their TL sequence, to redo the clustering and try to identify what will be the best way to cluster this data and to obtain a better quality of the data. So when she did that, she looked at 2 parameters with the size of the cluster, but also the SI. The SI is basically the entropy of the cluster, or if you want, an estimate of the fact that it's more peaky or more broad. Okay, and so with this, you obtain clearly 2 type of clusters, as you can see here, and it has been validated by this statistical assays. We have to have 2 type of assay, one we call sharp and one we call broad. And actually the sharp are associated with the data box, when the broad are not associated with the data box. And the sharp cluster and data box containing genes are more prone to be regulated, and we show that by using our RNA-seq data. And what was interesting is that this dichotomy between data box containing a cluster and broad and non-data box containing cluster, broad and sharp, have been observed exactly the same way in Saccharomyces cerevisiae and in other species, suggesting that our new way of clustering the TSS was fine and was provide some Guilhem clusters. So now we did that for the full genome. I identified 3,299 alternative TSS clusters associated with nearly 2,000 genes. It's not the number of clusters, it's number of alternative clusters, right? So she classified them in 3 categories: the one that change the size of the TL sequence of the 5' UTRs, the one that are very close downstream to the ATG that basically change N-terminal of the protein, and some others that are very downstream of the genes that are supposed to promote the production of a much shorter RNA. And because we don't know the function of these things, of this RNA, we call them transcription resulting from alternative start sites, although we have no idea of the function they might have. So the first category are the ones that are associated with the TL sequence, and I come back to my model of translation here. And I told you that if there is a new off, then the translation machinery will stop, translate the UOF, and the mRNA will be degraded to the ND pathway. And I remind you that the work from Edward Wallace, when he was in my lab, showed that the more UOF you have, the less the transcription efficiency is. And it also showed that the gene that contain UOF are more prone to be controlled by the NMD pathway that was going in the right direction. So now, if you imagine that there is an alternative TSS that shortened the transcript leader sequence and basically skipped the UOF, then this new short mRNA should be immune of the NMD. And exactly what we found. We found that genes that contain UOF and that are and contain also alternative TSS are less prone to be regulated than the one that contain UOF without alternative TSS. And we found a very nice example of this type of regulation. In this case, when you delete UPF1, which is a major gene necessary for NMD, you see that you increase basically the appearance of the size of the coverage, because you reveal the presence of an alternative TSS that is upstream here, and when it's used, produce an mRNA that contains a UOF that is sensitive to the NMD pathway. So basically, the alternative TSS is regulating the gene expression by skipping or avoiding the NMD pathway. The second consequence of the alternative TSS usage is changing the N-terminal sequence of the protein. And I show you here two examples of alternative TSS. Here, those two clusters are very close, but the second one is too close. So the ATG that is here is not used. And so the material targeting motif that is here is will not be used. So basically the alternative protein will start here and will be targeted to the cytoplasm, whether the full protein will be targeted to the mitochondria. In this case, it is the same. You have two alternative TSS, and the second one will promote the protein that goes to the nuclear, when the first one will target the protein that goes to the mitochondria. We did that systematically. She observed different possible targeting, and to validate this alternative targeting, she used a gene named ME102 that is regulated by alternative TSS. And you see that they have a TSS that is mostly used in exponential phase at 30 degree that basically skip the mitochondrial targeting signals when in stationary phase. The long RNA is produced and supposed to produce a protein that it will be targeted to mitochondria. So we tagged the protein with MNEO in C-terminal and looked on the microscope. And indeed, what she found is in the wild type, the protein in stationary phase go to the mitochondria, or apparently go to the mitochondria, because the long mRNA is produced, so long protein is doing. When in exponential phase, when the short protein is doing, the protein goes to the cytoplasm or to the nucleus. When you eliminate the translation star site, the first translation star site, then you maintain the localization in exponential phase, but you lose the localization in mitochondria in stationary phase. And the same if you eliminate completely the mitochondrial targeting signals. So showing that alternative TSS regulate protein subcellular localization. Finally, we have alternative TSS within the earth, and there is a lot of them, and they seem to produce alternative long coding RNAs or alternative mRNAs. So some are conserved. You see that this example, we have the same position in Cryptococcus neoformans and Cryptococcus denoformans. Some exist in both species, but not exactly in the same position. Okay, to try to see whether some can be translated, we perform N-terminomics. So it's basically we sequence only the beginning or the N-terminal sequence of each of the proteins. We did that we obtain 844 N-terminal peptides with the help of Chiara Guerrera from Necker Paris, and 818 were associated with a classical annotate protein, so N-terminal sequence of the annotate protein. 19 when N-terminal sequence of alternative protein associates with the usage of alternative CSS, so the one that are very close to the annotate IgG, just the one that I just spoke about. But 7 N-terminal sequence of alternative protein associated with the usage of an alternative CSS that is far downstream to the IgG, suggesting that at least some of them are translated in some protein and some peptide, the function of which is unknown. This alternative TSS is regulated by the growth condition. It's we compare this in 4 different conditions. We identify 2,520 TSS usage regulation in 627 genes. This regulation seems to be specific of the comparison we make. I mean, 25% of this regulation seems to be specific, suggesting that indeed there is a specific regulation and specific mechanism to regulate alternative TSS. So overall, we have alternative CSS that regulate the length of the 5' UTR and skipping or not skipping the UOF and regulating the expression of the gene. We have alternative CSS that regulate the targeting of the protein, and we have alternative CSS that regulate the production of a stress that we don't know the function of. So the next step was to understand the mechanism by which those alternative CSS were regulated, and we start with a very simple hypothesis, imagining that in one condition there will be a transcription factor that will promote, for exampt, the transcript of a long mRNA, and in another condition, another transcription factor will bind downstream and promote the transcription of shorter mRNA. And she used to validate a model, a collection of mutants produced by Sunban in Korea, where they delete all transcription factor in Cryptococcus neoformans. Use also model genes these PKP1 genes, for which there's two alternative CSS, one that is mostly used in stationary phase and one that is used both in exponential phase and in stationary phase. And she used a qPCR assay and showed that indeed in exponential phase and in stationary phase, the long and the short mRNA have about the same level of expression, where in stationary phase, the short isoform is much more expressed than the long one. And then she extracted the RNA from all those mutants into two conditions, and she did RT-qPCR assay for each of them to identify one single mutant that she named TER1 for TSS usage regulators. And you can see that in the TER1 mutant, we have the short version of PKP1 is not affected in stationary phase. The long version is not affected, but the short version is less expressed than in the mutant than in the wild type. And this modification is not observed in the wild type, in exponential phase, suggesting that TER1 is indeed regulating the expression of the short isoform without affecting the expression of the long one. TER1 seems to be a more general regulator, actually. V produced TSS-seq for the TER1 mutant in exponential phase and in stationary phase, and she looked at the regulation or the impact of this mutation on the regulation of alternative TSS in all the genes in Cryptococcus. And of course, because we have much less gene to be observed, because we have to compare many conditions and have enough data for all the data, for all the conditions, she was only able to compare this data for roughly 180 genes. But still, she showed that the regulation we observe in wild type for these 180 genes, most of them were lost in the TER1. So in TER1, all the regulation is lost. For example, the PKP1, we knew about it, and it was apparent from the screen. It was pretty prevalent in the wild type and completely lost in the mutant. It was lost also for EDP1. For SOD1, there is no modification of the pattern, and the impact of the deletion of TER1 does not impact alternative CSS. What was interesting in some case, like in YCS1, we observe a new regulation that is dependent on the absence of TER1. When TER1 is not there, when we see this new TSS here that is not present in the wild type, and its TSS is regulated by the phase of Rose. So of course we come back with MA2 and look whether MA102 was also impacted by the deletion of TER1, and indeed it was. Remember, in stationary phase we have only the long one, in exponential phase we have mostly the short one, and in the TER1 mutant we lose completely this regulation, and exactly what we see when we looked at the localization of the protein in the wild type is cytoplasmic and nuclear in the exponential phase, and mostly mitochondrial in stationary phase, where in TER1 this localization is nearly lost in the exponential phase, and it's very blurry or mixed in stationary phase, and that's been confirmed also by a qRT-PCR. TER1 have been associated with virulence by Sunban and in multi-infection assays, so we didn't do it again, but we just, because it's a mitochondrial protein, because it regulates the expression of mitochondrial proteins, we looked at the impact of TER1 deletion in meningeal and antimycin resistance, basically in oxidative stress resistance, and we found that there is a modification. In collaboration with Robin May, we also showed that TER1, the mutant, were more easily phagocyted than the wild type, suggesting and confirming the impact of TER1 and probably alternative CSS usage on virulence and host-pathogen interaction. Overall, from there, we know that alternative CSH is very prominent in Cryptococcus, and there is, since our paper last summer, there is additional paper that shows that in Cryptococcus. There's a fantastic paper from Sharon Lin actually showing this and show how the alternative CSS regulate filamentation in Cryptococcus. Alternative CSS regulate gene expression as well as transcriptome and protein structure. it's a major regulator of alternative CSS during exponential phase and stationary phase, suggesting like a program a program regulation of transcriptome modification, in addition to the classical one observed by RNA-seq and comparing gene expression. And we show that third one is regulate resistance to oxidative stress and virulence in Cryptococcus. If I come back to the model of gene I got at the beginning, I'll show you that in Cryptococcus there is an annotated ATG, some downstream ATGs and upstream ATGs, and the usage of these different translation codon regulate the proteome diversity and mRNA expression. I'll show you that alternative TSS will change this pattern and regulate as well mRNA stability, protein targeting, and probably long-coding RNA or alternative proteins. And we can imagine additional mechanism like, for example, alternate splicing that can skip the U-off and maybe regulate mRNA stability, or alternative polydylation sites that will change the size of the 3' UTR or the size of the protein, or producing long-coding RNA of as well alternative proteins. Before finishing, I would like to thank the member of my lab, and mostly Edward Wallace and Vidang that did most of this work, Chiara Guerrera, Robin May, and Ethan Malani for collaboration. Thank you very much.
Fantastic. Thanks very much, Guilhem. We're going to go on to the question and answer period, and Geraldine's going to lead off.
Okay, so can I just remind everybody to keep putting the questions in the Q&A with the initials or the name of the speaker. Right, so Rebecca, we have a lot of questions for you. The first one, have you looked at the association between activated expression and the fold change enrichment for genes where you have several guides, several hits? Have you enough data to do that?
A great question. We haven't for our current hits. I showed, previous data where we've done sort of like genotype correlations of individual genes, but we haven't yet from hits from the pool. And I think that would be great to start off with. We would have to actually do like individual QRT-PCR on all the strains, because we don't have prior information about what the level of expression of all of our genes in our pool are. But yeah, that's something we're interested in.
Okay. Question for Guilhem from Ted White. Very nice talk and very interesting. 2 questions. Are upstream regions conserved in other fungi for the same gene? And Matt Sachs has done work on upstream translation regulating expression levels of the downstream gene. Do you have any evidence of that?
For the conservation, I've not compared systematically the conservation of U-off position. Is kind of difficult because the sequence of the 5' UTR are not very well conserved. And so, and because most of the gene contain U-off, basically you're going to have a large overlap with this. I'm not sure it will be really meaningful in that sense. For the second question, what was that exactly?
Matt Sachs has done work on upstream translation regulating expression levels of downstream genes. Do you see evidence of that?
Well, if I understand, the idea is that Matt look at how the upstream ATG will regulate the expression of the downstream ATG, right?
Exactly.
So the thing we have is exactly what we found, is that there is the Kozak motif, whether it's strong or not, will impact the expression of the downstream one. Whether it is the mechanism by which it's working The mechanism by which those quality of motifs are recognized, we have not studied them in Cryptococcus. They have been studied in some other settings, and including by Matt, but we have not done that in Cryptococcus.
Okay, back to Geraldine.
Okay. So Rebecca, for the non-genomics people, could you explain how you count the number of chromosomes?
Oh, we don't count the number of chromosomes, or I guess we haven't for any of the work we've done. I guess maybe in referring to like the trisomy work. Yes, right. Okay. So that work, the stuff I showed, none of that was ours, but it is done through sequencing and basically like assembly of mostly short reads and looking at relative counts. So it's where we see, you know, like a proportional increase in the number of, like, you know, 50% increase, I guess, in the number of reads across.
Yeah. Okay, thanks, Rebecca. Joe?
Okay, Guilhem, a question from Matt Blango. And this is a two-part question. We'll do part one first. I'm wondering if you see any RNA secondary structure conservation by prediction in the leaders when comparing neoformans and d-neoformans, which might inform about regulatory elements.
Okay, that's what we have not done. It's actually, I suppressed this part for my presentation because something that is a certain level of regulation between the usage of the first and the second ATG is done by indeed the position of the ATG. The second one is indeed the COSAC, and the third one is the presence or the regulation by by a second structure within the TL sequence. We have not done that, and I'm not sure that. I mean, in my point of view, it's very difficult. It's not difficult to predict. It's very difficult to validate their presence. And so I don't know how, but we have not done it at all. And if we have some people have good tools to do it, I would love to do it.
Okay, and then the second part of the question is, any hints from the leader data as to what transcripts might ultimately end up in extracellular vesicles?
Okay, we sequence the transcriptome of the extracellular vesicles, and right now we sequence the transcriptome that is outside of the cell. Right now we have no clear experiment done to show that those RNA we found are associated really with extracellular vesicle, whether they are inside, at the surface, or outside, completely unrelated to that. So I will be very cautious in what I'm going to say, but let's say we have not found very spectacular data about concerning the presence of alternative transcript in the associated with the extracellular transcriptome.
Okay, thank you. Back to Geraldine.
Okay, Rebecca, an interesting question from Ted White. So chromosome R has the rDNA locus on it. So have you looked at the copy number or the expression of that?
Yeah, that's a great question. And everyone is reminded of that because it's called chromosome R. Otherwise, nobody would remember. But yeah, so we haven't overexpressed the R DNA. We would really like to and will attempt to. The graduate student who's working on this was just reminding me there's a couple sort of like technical reasons why we're not sure the system will work. We don't have great assemblies of the rRNA is sort of part of it. And then we're not sure that the CRISPR-A system, that the activators that we use for the system will work with RNA polymerase 3. So we're not, and I think that hasn't been shown in other species. I'd have to look into that. But so I don't think we have evidence that the system would necessarily work well. But it'd be nice to think about other ways that we could overexpress those as well.
Yeah, my guess is it'd be quite complicated to do that. Maybe I'll ask you one more. So from Christopher Zajac, the multiplexed Cas12 system, so where you've got several guides in the array, is there any evidence that it matters which order they're in? So will one be used more than the other?
Yeah, that's a great question. And there is evidence of this in the literature. So there is suggestion that initial guides will be used preferentially compared to others, but there's all sorts of other kind of parameters and what makes for sort of a good, efficient guide. And so that's something that we're actively looking into. What Nick, the student who's been working on this project, has been doing is putting in a number of sort of non-targeting guides. So, if we have a guide targeting like, CDR1, which we've shown can overexpress, putting it sort of first, or putting like a blank guide and then putting it second, or then blank, blank, and then third, and then blank, blank, blank. And so we're seeing sort of how far we can push it down the guide array and still have it expressed to comparable levels. And so far, it actually doesn't seem to be a big concern. I don't know how far we've pushed it down. But that's something that we're considering, and we can monitor, obviously with like QRT-PCR to make sure that we're getting kind of consistent activation.
Okay. Do you have any more questions, Joe?
I do. Guilhem, there's a question about the TER1 factor that you identified that's involved in regulating alternative transcription start sites. And what does TER1 look like? Is that a DNA binding protein or an RNA binding protein? You're muted.
Its original name is FCY27. FZC 27, so is a zinc finger transition factor, and it looked like a DNA binding protein. So we have not proved that. That's one thing that's actually we at one point we're thinking of doing, is tagging the protein to show that it was going indeed to the nucleus, but it's still possible that it's an RNA binding protein, although it's predicted to be a DNA binding protein.
I wonder if you could do something like ChIP-seq to see if it's
so we've done this. We've done ChIP-seq. We've done even DAM-seq, DAM-ID-seq. We have not found any TER1 was binding to the we produce peaks. Okay, so we obtain peaks that suggest that it goes indeed to the nucleus, and that it binds to the nucleus. But if you look at the pattern of TER1 by ChIP-seq or by DAM-ID-seq, it's very difficult to conclude about any mechanism by which the regulation is made. So the regulation might be completely indirect, or at least we have not found any specific locus at which we can explain the expression of the usage of TSS just by the binding of TER1.
Okay. All right, back to Geraldine.
Okay, Rebecca, I'm going to ask you two again. So firstly, can you tune the Cas9 system by making either a Cas9 or a guide RNA inducible?
Yes.
Okay.
The work that I showed isn't using an inducible system, but we have an inducible system for our CRISPR interference system. We were sort of more concerned about knocking down genes, because if you're turning down an essential gene, you might lose it from your pool before you even have a chance to study it. So we made sure that one was regulatable. And we didn't initially with the activation system, but we are now.
Okay. And do you have any idea now what guide RNAs work now that you've tried 6 per gene for a lot of genes?
Yes, these are all the questions that we're asking ourselves. Yeah, yes, we are trying to learn both from this pool, and we have other CRISPR-I pools that we've developed, some very large and some that we've screened at like high, under like many different conditions. So the more guides we have, the more screening conditions we have, the more we're learning about which guides work. And so that's exactly what we're doing. And that will allow us to sort of determine better what makes good guide efficiency. And we're going to continue to sort of pare down the pools as we learn that.
Another interesting one from Lindsay Tolhurst. Can you activate genes that are completely off their silenced, like in heterochromatin?
Yeah, so We haven't monitored this directly ourselves, but the literature suggests yes. So like other CRISPR-A systems in like mammalian cells, et cetera, have definitely shown that you can activate even like quite highly repressed heterochromatin loci.
Okay.
Yeah.
Over to Joe.
Okay, Guilhem, a question about the comparative analysis. So you've compared 3 different Cryptococcus species, neoformans, D-neoformans, and Deuterogatii. And the first 2 have RNAi, and the third one lost all of its RNAi pathway. And do you see any differences in the strains that are RNAi proficient versus the deficient one?
So we have not done this this work on TSS was done only in neoformans and D-neoformans. We have not done that in Deuterogatii. We have done some annotations of the genome of Deuterogatii using only RNA-seq, and we found some apparent regulation of atenazy splicing or antron retention associated with the absence of RNAi in there, but we don't know exactly what it means, and it's difficult to be sure that it's related to this or to the fact that this is just a different strain.
Okay, so maybe a good opportunity to do some more transcription start site and polyadenylation analysis. Back to Geraldine.
Hey, Rebecca, a long question from Alex. So I think the gist of it is that you've shown that overexpression of SEC4 increases tolerance, I think it was. So that's a RAB GTPase. And it's kind of surprising that overexpressing one part of a secretory pipeline would have a dramatic effect if all the other parts are not. So what do you think is happening? Is it very dynamic activity and recycling back to the Golgi? And then a very specific question, is the hyphal elongation rate increased in your overexpression strain?
Yeah, that's a super cool question. I guess the short answer to the second part is we haven't looked at hyphal elongation, but that's a great idea, and we totally can and will. And I don't know. I agree that it would be sort of surprising that overexpression of sort of a single factor would be sufficient or would allow for maybe increase in the like secretory system without others. Now, yeah, so I agree. I don't know, I guess, is the short answer for what exact kind of connection between overexpressing this RAB GTPase and the increased tolerance that we see is. We certainly know from the Saccharomyces literature that there is all sorts of, or at least like deletion, or I think it's essential, so like downregulation of SEC4 has all sorts of phenotypes involving like cell cycle and autophagy and all sorts of processes. So I'm not sure yet what's happening, but
I think we're just about out of time, and there are 2 more questions, so see if you could handle them very briefly. First is, are the examples where the dosage is more important? So does it matter whether you activate or repress, you get the same phenotype? And secondly, does population heterogeneity impact azole resistance?
Yes, okay, I'm also like reading the questions. Yeah, so for the first question, Alicia, right, are there examples where increase and decrease of 2 genes together is leading to resistance like changes? I don't know. That's a great question. I'm not sure. So I guess this is my short answer to that. And yeah, and population heterogeneity. So, right, so we do sometimes wonder about the effect of how strains are behaving in the pool versus individually. And there certainly are instances where we find something that has a really profound sort of phenotype. It changes significantly in relative abundance in the pool, and then we pull it out and it doesn't behave at all like we expected. Now, there's going to be some false positives, so it could be that. But we certainly might expect that in a pool of overexpression mutants, there might be some other interesting like metabolites that are being produced or something because of overexpression of various pathways that don't happen in an individual strain. So there might definitely be some differences there, and that's something that we're thinking about.
Okay. Yeah, right at a time. My office lights
Yeah.
went out to remind me of that. Okay. So we're right at the end. Thank you very much, Guillaume and Rebecca. They were fantastic talks. And thanks to my co-chair, Joe Heitman, and to everybody who attended. And goodbye for now.
Thank you. Bye-bye.
Thank you very much. Have a
Thanks.
good rest of your days.